Hello, everyone, and welcome. My name is Runa Deeb. I'm a senior principal at Geosyntec Consultants in Oakland, California, and the coordinator of the webinar series on behalf of SERDP and ESTCP. I have the pleasure of facilitating today's event. This webinar focuses on DoD-funded research efforts to assess ecological PFAS exposure and effects. First, Dr. David Moore from the U.S. Army Engineer Research and Development Center will present results from a multi-generational study of the effects of low-level PFOS exposure on zebrafish. Dave's presentation will be followed by a brief Q&A session. Then Dr. Christine Custer from the USGS will present research into the potential effects of PFAS on a model avian species. Chris's presentation will also be followed by a brief Q&A session, and we will conclude the webinar with a longer Q&A session featuring both of today's speakers. The next few slides provide instructions on optimizing your webinar experience. If you have not done so already, please download Zoom at the link shown here and provide it to you in your webinar registration confirmation email. If you're unable to download Zoom, you can view the slides using a compatible internet browser such as Firefox, IE, or Edge, and by creating a free Zoom account. If you're unable to view the slides, or if your screen freezes, try keying in Control and F5 to perform a hard refresh of your browser. If you're accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to have difficulties, call into the conference line shown here. You can also submit a comment using the chat box asking for assistance. Just remember to use the chat box only for comments related to technical difficulties. The Q&A option should be reserved for questions for the speakers. And in case of continued technical difficulties, you can download a PDF of the slides from the webinar webpage and call into the conference line. Note that we will also be live streaming the webinar on the SERDP and ESTCP YouTube channel at the link shown here. So that's another way you can follow today's proceedings. The broadcast will be listen only. You may submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A period to submit your questions. We do encourage you to get them in as soon as you're able to. And when you submit your questions, please add your organization name at the end of the question so that we can identify it. With that, I'd like to introduce Cara Patton, who is an environmental project manager at Nobles and the technical assistant for SERDP and ESTCP's environmental restoration program area. Cara has worked with SERDP and ESTCP since 2008, and before that, she worked at the Drug Enforcement Administration, where she was a forensic chemist. Cara received her master's degree in chemistry from George Mason University. Cara, please get us started. Thank you.
Great, thank you, Rula. I'm happy to welcome everyone to today's SERDP and ESTCP webinar. The next few slides will provide a quick overview of both SERDP and ESTCP. SERDP is the Strategic Environmental Research and Development Program. It was established in 1991 by Congress as a partnership between the Department of Defense, the Department of Energy, and the EPA. SERDP's mission is to identify and address high-priority environmental science and technology opportunities that focus on DoD requirements. SERDP funds both fundamental research as well as advanced technology development that ultimately impact real-world environmental management. ESTCP is the Environmental Security Technology Certification Program, in which we demonstrate innovative environmental and energy technologies. These investments capitalize on past investments under SERDP or other research programs and are designed to transition technologies out of the laboratory and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and regulatory acceptance. SERDP and ESTCP are complementary programs with much of SERDP research occurring at the lab and pilot scale with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although some occasionally include supporting lab efforts which are conducted. There are several environmental drivers for the type of work funded by SERDP and ESTCP, with the underlying objective of sustaining DoD ranges, facilities, and operations. This, as you can imagine, is a broad undertaking. It takes the form of looking at maritime sustainability, threatened and endangered species, climate impacts, unexploded ordnance and munition constituents, as well as other environmental drivers. One key environmental driver is the reduction of current and future environmental liabilities. This involves addressing contamination from past practices, including impacts to groundwater, soils, and sediments, UXO contamination, and developing management approaches for contaminants of emerging concern. The second part of this is pollution prevention, which is a focus on eliminating chemicals of concern or hazardous materials in manufacturing, maintenance, and operations on our installations. We have several main focus areas for research and demonstration at SERDP and ESTCP, as shown on this slide here. SERDP and ESTCP are funding a broad range of PFAS-related projects. You can access an overview of our PFAS program as well as project descriptions on our website. The boxes on this slide along the top of the graph illustrate workshops that have been held to develop a strategic plan for addressing PFAS issues. A summary report was prepared for each workshop to summarize the discussions and to identify research, demonstration, and technology transfer needs. To view workshop reports, click associated boxes at the top. Each SERDP project has an associated box with a project title, which links to a full description of the project, as well as any reports that have been published as part of the project. Each ESTCP topic listed represents a single topic. Click the project title to go to the project webpage. As mentioned earlier, technology transfer is very important within the SERDP and ESTCP programs. Technology transfer efforts include the development of videos, training workshops, and guidance documents. The webinar series is a substantial component of our technology transfer efforts. Our webinar series highlights research and demonstration efforts from all of our program areas. Upcoming topics include coding biodegradation and prevention, autonomous underwater vehicle-based structural acoustic sonars for underwater buried UXO detection and classification, the effects of phenology on species of management concern on DoD lands, and others. The next environmental restoration webinar is on August 10th on the removal of munitions constituents from wastewater. Registration is open for webinars through the end of this year. You can find additional information about upcoming webinars at this link. All past webinars are archived and can be accessed using this link as well. I would like to remind you that a copy of the presentation of today's session can be downloaded from our webinar webpage. We would appreciate it if you can please take a moment to complete the survey that will pop up on your screen at the end of the webcast. I hope you enjoy the webinar today. And finally, please save the date for the 2023 symposium, which will be held between Tuesday, November 28th and Friday, December 1st in Arlington, Virginia.
Thank you so much, Cara. At this stage, I'd like to introduce our first speaker, Dr. David Moore, who is a senior research biologist with the U.S. Army Engineer Research and Development Center in Vicksburg, Mississippi. Dave currently leads ERDC's program on advanced materials and substances of emerging environmental concern. He has 35 years of experience and has served as principal or co-principal investigator on projects funded by SERDP, the Office of the Secretary of Defense, the U.S. Navy, as well as several industrial sponsors on topics relating to PFAS, AFFF replacement products, and the application of novel modeling approaches for the risk characterization of PFAS. Dave has authored over 40 peer-reviewed publications. He received a bachelor's degree in biology from Washington and D University and a PhD in environmental toxicology from the University of South Carolina. David, we're very happy to have you. Please proceed.
Well, thank you, Rula, and thank you, Cara, for the introduction and the opportunity to speak to the group today. I'll be presenting results from a study that we did that was a multi-generational exposure of the zebrafish to PFOS. And before I begin, I think it'd probably be helpful to provide a little bit of background for the genesis of this study. As everybody is aware, there are a number of organizations, including EPA and DoD, that are actively engaged in assessing and addressing potential ecological and human health risk from environmental exposure to a variety of PFAS. One of the high priorities is development of environmental exposure threshold effect concentrations in water, sediment, and tissues. Typically, the values that are derived are using results from peer-reviewed publication. In 2016, the Environmental Protection Authority of Victoria, Australia released water quality guidelines for PFOS that included a freshwater 99% species protection level of 0.00023 micrograms per liter. That value is essentially 100 to 1,000-fold lower than most of the other regulatory levels that we have out there currently. Derivation of this low threshold was driven primarily by results of a single study conducted by Keeter et al. In 2012. It was a multi-generational exposure of the zebrafish Danio to PFOS, and it reported reduced growth at a concentration of 0.6 micrograms per liter. There were a number of issues associated with the Keeter et al. Study. It wasn't really designed or intended to be used for the derivation of regulatory thresholds. It had low replication, a low number of widely separated exposure concentrations, and it wasn't entirely clear the statistical analysis that was applied that created a lot of uncertainty around the use of this study for the derivation of these thresholds. So with that, I'll If I could go back to slide 21, please. So with that, I'm going to start in on the presentation and give you just sort of an overview of the presentation. We'll start by describing our technical objective, which relates to this Keeter et al. Study. We'll describe the approach. Then we'll step through the results from the study, and finally end with conclusions and describe some of the benefits to DoD. 22. So the technical objective for the study was basically to repeat the Keeter et al. 2012 study using a more robust experimental design to reduce uncertainty of the results. We met in 2019 with technical reps from industry as well as representatives from EPA ORD and EPA headquarters to discuss the experimental design and reach consensus on the approach. And I'll talk in more detail over the next couple of slides. In addition, there was an opportunity to leverage this study to provide additional endpoints and additional test species and protocols to include the support for development of an adverse outcome pathway and parameterization of computational models, enable comparison to other sensitive species, and also develop toxicity reference values to support risk assessments. '23, please. So conceptually, the Keeter et al. Study, as I mentioned, only included 3 PFOS concentrations and a control, and only included 2 replicates tanks per treatment. 24. In the ERDIC design that was developed in consultation with both EPA and industry, we increased the number of concentrations from 3 to 5 we increased the replication from 2 to 5. We increased the number of fish per replicate from 30 that was used in the Keter et al. Study to 50. And we provided for separation of the breeding groups. And the design was based on a calculated statistical power of 0.8. 25, please. So in terms of the endpoints, we measured survival. That was measured on a daily basis. Growth was measured as both wet weight, length, and area, and measurements were collected at 30 days post-fertilization, 60 days post-fertilization, 90 days post-fertilization, and 180 days post-fertilization. Beginning at about 4 months, we conducted a series of reproductive trials. Reproduction was conducted 8 times for both the parental as well as the F1 generation. And then at the end of each generation, we collected animals for subsequent analyses of RNA sequencing, vitellogenin, as well as histopathology. And finally, we also collected tissue residue analysis. I won't be presenting the results here today as part of this presentation, but I will describe some of those results in our conclusions. We collected tissue residues in the parental generation at 14 days, 29 days, 180 days, and then in the F1 generation at 180 days. Slide 26, please. So this is just a conceptual diagram of how the study was conducted. On the left-hand side of the slide, you'll see the parental generation's exposures for 180 days, beginning with fertilized embryos. Those embryos were exposed under static conditions for 14 days. And then when we got to the swim up stage, the animals were transitioned over to the tanks and exposed under static renewal conditions with daily renewal of exposure water. And then at the end of 30 days post-fertilization, the animals were then transferred in the tanks to a flow-through system and exposed through 180 days. As I mentioned, beginning around 4 months or around 110 or 111 days, when we could discriminate between males and females, we culled from the original 50 animals per tank down to 15 males and 15 females, and then conducted a series of breeding trials over the course of the remaining 8 weeks. The final reproductive trial, the eggs were obtained, fertilized embryos were obtained from that. Again, we repeated the process. Those were exposed under static conditions for 14 days, transitioned over to tanks, exposed under static renewal conditions up to 30 days post-fertilization, then transitioned to the racks and exposed up through adulthood under flow-through conditions. And again, at approximately 4 months, we culled the animals from the initial 50 animals per tank down to 15 males and 15 females for the subsequent breeding trials. And then at the end of the breeding trials, we again collected fertilized embryos, exposed those under static conditions through 16 days, and collected measurements of both survival and developmental status on the embryos in the F2 generation. Slide 27, please. This is just a schematic showing our exposure system. In the lower right-hand side of the photograph, you can see a bunch of brown carboys. These contained concentrated stock solutions. The concentrated stock solutions were then moved by peristaltic pump into a mixing chamber at the head of each exposure chamber, and to achieve the desired exposure concentrations. Slide 28, please. So one of the issues we encountered, we did a lot of background measurements of materials associated with the exposure system. We measured PFAS in the food that we're going to be frying, the organisms. We measured our laboratory water, et cetera, et cetera. And one of the issues we found is that our highly polished laboratory water system actually contained measurable levels of PFOS, largely through Teflon fittings and those kinds of things that are associated with those types of systems. So we actually had to pre-polish our water system prior to dosing the PFOS by passing it through purifying resin to remove any residual PFAS prior to generating our exposure concentrations. On the back end, water was collected in this white equilibration tank that you can see over on the left-hand side at the base of the flow-through system. That water was then in turn passed through a particulate filter, activated carbon filter, and then through 3 canisters of purifying resin prior to being discharged to a holding tank outside. We generated about 55 gallons of exposure water waste each day, so quite a bit of water. That water was held in a frac tank, and then we measured that to ensure that there was no measurable quantities of PFOS in the treated waste stream prior to discharge. Slide 29, please. As I mentioned, in addition to the multi-gen study, we also ran a series of more standardized tests. Included the 30-day chronic OECD method with Danny O'Rero, measuring survival and growth. We also conducted a 7-day chronic study with the water flea Daphnia dubia, a 42-day chronic study with Hyalella azteca, and a 20-day chronic study with the midge Gronomus dilutus. Slide 30, please. So now getting into how we monitored our exposures. Our PFOS concentrations were measured using liquid chromatography with tandem mass spec. We sampled 2 random tanks at each target concentration weekly through the first 4 weeks of the exposure, and then every other week through 180 days for each of the parental and F1 generations. In the subsequent figures that I'll be showing in the results, where we're looking at exposure concentrations associated with endpoints, it should be noted that the ecotox endpoints are reported as a cumulative mean concentration to the day of the sampling endpoint. So, for example, a 30-day growth endpoint would be the exposure concentration associated with that would be the time-weighted average of the exposure concentrations through day 30. Slide 31, please. So on the right hand of this slide, you can see the measured exposure versus nominal concentrations for the parental F1 and F2 generations. The parental generation exposure concentrations is in the top panel, the F1 generation is in the middle panel, and the F2 generation is in the bottom panel. The target PFOS concentrations, the nominal concentrations are listed across in the x-axis, and the measured concentrations are on the y-axis. In both the parental generation and the F2 generation, our measured concentrations were very close to nominal. In the F1 generation, we were slightly reduced relative to our target nominal concentration. We had a period of about 3 weeks where the concentrations, the measured concentrations were reduced relative to target, and we went through a variety of forensic analyses to try to establish what was happening. We measured both in those brown carboys I showed earlier to ensure that we were setting up the stock concentrations appropriately. We measured in the mixing chambers. All of that seemed to align with we were getting the appropriate concentrations into the system, but something was happening within the system that we weren't able to account for. So we essentially increased the stock concentrations to account for that diminution, and we're able to get the exposure concentrations back online. But for that period of about 3 weeks, it reduced the overall average concentration somewhat relative to what we experienced in the F1 and I mean in the parental and the F2 generations. If I could have slide 32, please. So this slide shows the effects on survival. It's a lot of data. So again, in keeping with the theme, the top panel is the parental generation, the middle panel is the F1 generation, and the bottom panel is the F2 generation. The colorations moving from left to right are control to the darkest bar represents the 100 ppb concentration. And then the measurement endpoints for the parental and F1 are at 10, 15, 20, 30, 60, 90, 111, and 180 days post-fertilization. The take-home message from this is we saw reduced survival at the highest concentration in both the parental and the F2 generation, And indeed, though it's not shown here, subsequent statistical analyses done by EPA, hot off the press last week, shows that the highest concentration is in fact statistically significant. And so we did not see this in the F1 generation, but as I alluded to in the previous slide, the overall exposure concentrations were slightly reduced relative to the target in the F1 generation. So as you remember, the top end concentration in the F1 generation was 75 micrograms per liter versus the closer to 100 that we achieved in the parental and the F2 generation. So that could potentially explain why we're not seeing in the F1. Slide 33, please. Now moving to growth, this is growth expressed as wet weight. One of the issues we ran into is because of effects on survival, we didn't adjust ration based on the number of fish in each tank. And so we noticed in analyzing the data that in those tanks where there was reduced survival, the fish were actually a little larger, and so more resources for fewer fish. And so in discussions with EPA, it was determined that we would move our growth endpoint to a combined endpoint of biomass per replicate, so that is utilizing the weight information along with number of animals in each tank to generate a biomass measurement. And looking at biomass, again, here we have in the top panel, this is the parental generation. Middle panel is the F1 generation. The bottom panel is the F2 generation. The figures A through C represent 60, 90, and 180 day weight measurements. And as you can see, what we see is with increasing exposure concentration, we did observe a negative effect at day 60 in the parental generation at the highest concentration. In the F1 generation, we saw some diminution in growth at day 90 in the 0.56 to 2.97 in the 89 part per billion concentrations. And in the F2, we saw no significant effects. Slide 34, please. Moving to measurements on length. The only, again, figures are being presented in similar fashion across the next series of slides. So in the top panel represents the parental generation, the middle panel represents the F1 generation, the bottom panel represents the F2 generation. Again, 60, 90, that should say 180 days in figure C instead of 90. But 60, 90, and 180 moving from left to right, and then 16 days for the F2 generation. The only significant negative effect that we saw on length was male body lengths at 180 days in the two highest exposure concentrations. In the F1 generation, we saw no significant differences relative to controls at either 60 or 90 days. But again, male lengths were significantly reduced at 180 days in the highest exposure concentration. For the F2 generation, we did see significantly reduced growth or length in the highest concentration at 16 days. I can have slide 35, please. Moving on to reproductive effects. In both the parental and the F1 generation, we measured mean number of eggs per female as well as viability of those eggs. And we saw no significant negative effect for PFOS on egg production or egg viability across the 8 reproductive trials in either the parental or the F1 generation. Moving on to slide 36. Some prior studies had shown estrogenic effects associated with PFOS, and so we wanted to evaluate that. And in order to do that, we collected whole adult male zebrafish at 180 days from both the parental and F1 generations, and we assayed them for vitellogenin to determine whether we were seeing any evidence of estrogenic effects. We collected 8 males from each concentration for each generation and assayed them. And we also collected control females from each generation and used those as positive controls. The bottom line from this analysis indicates that there's no significant induction of vitellogenin in male zebrafish in either the parental or the F1 generations compared to the controls. And the values of vitellogenin production was, as expected, very low. Slide 37, please. When we were doing dissections at 180 days in both parental and F1 generations, we noted fatty livers in male zebrafish in the highest exposure concentration. So on the basis of that, we made a determination to have a confirmatory histopathology performed by EPA. It is known that the liver is a target organ of PFOS exposure in fish and other animals, and PFOS exposure can lead to liver steatosis. Keter noted this in their study. As I mentioned, we collected a minimum of 8 male liver samples per concentration at 180 days, and we analyzed in both the parental and F1 generations the control, the 20 ppb and the 100 ppb. And we did, in fact, see evidence, if we'll move to the next slide, 38. We did see evidence of elevated hepatocellular evacuation in the 100 ppb treatments. The highest incidence and severity of this was in the F1 exposures, but although it was lower, we still saw a significant response in the parental generation when compared to the concurrent control responses. Now if we can move to slide 39, please. Adverse outcome pathway development and transcriptomics are important components of this work. There is an existing adverse outcome pathway for zebrafish on liver steatosis in response to PFOS exposure, and it's in preparation for submission to the AOP wiki site for continued development and peer review and publication. You can move to the next slide, please. So as I mentioned, we collected total RNA from the livers of males exposed to the control 0.6, the 3.2, 20, and 100 ppb concentrations of PFOS in both the parental and F1 generations. The RNA sequencing is complete. Walter Reed did the RNA sequencing for us, and we are currently in the process of doing the data analyses to determine whether PFOS treatments induced differential gene expression when compared to control fish. Slide 41, please. In terms of the more traditional type tests and species sensitivity comparisons, again, we ran studies with Chironomus dilutus, the midge, a 30-day growth endpoint with zebrafish, Dani Orero, 42-day reproductive test with Hyalella azteca, and a 7-day growth and reproduction study with Ceridaphe dubia. And you can see the color coding of the 4 species tested. Chironomus dilutus was the most sensitive, exhibiting the lowest effect thresholds of 0.0075 milligrams per liter for an LC50 and an EC10 for growth of 0.0015. This is consistent with what's been reported in the literature, as midges appear to be among the more sensitive freshwater species to PFOS. If I can move to slide 42, please. So to wrap things up here, our measured PFOS exposure concentrations in our flow-through system align fairly closely to the nominal targeted concentration, and were relatively stable and were generally confirmed by sample splits with Test America. We did have that one 30-day period in the course of the F2 generation where we had some reduced exposure concentrations, but we were able to correct that. In terms of survival, we saw no statistically significant effects on survival. Actually, I should correct myself based on the results of the analysis that was conducted last week. We did see statistically significant effects on survival in the highest exposure concentrations, that is 100 ppb, in both the parental and the F2 generations. Slide 43, please. In terms of growth, I mentioned that we had the confounding influence of food ration across the replicates, so we moved to a biomass endpoint, and we did see some indication of effect on this biomass endpoint at 100 ppb, concentration, but no real effects at 20 ppb and lower. And the 180 days, we did see some decrease in lengths in males, but not in females relative to controls at both the 20 ppb and the higher exposure concentration, but no corresponding effects on weights. In terms of reproduction, we didn't see any statistically significant effects on either egg production or viability relative to controls through the 8 reproductive trials that we conducted. Slide 44, please. No indication of vitellogenin effects in male fish, so no indication of estrogenic effects. For histopathology, we did observe fatty livers in male fish in the highest exposure concentrations in both the parental and F1 generation, and that was confirmed through histopathology. And as I mentioned on the transcriptional responses, that data is currently being evaluated. We do see some indication in that data of PPAR nuclear signaling in the embryos, and PPAR signaling is a critical controlling metabolic process, thus affecting bioenergetics. I can have slide 45, please. For the interspecies comparison that we did in conjunction with this study, it was determined that Chironomus tentans growth endpoint was more sensitive than the Danio-Raro 30-day growth endpoint, which was more sensitive than the 28-day reproductive endpoint for Hylola azteca, which in turn was more sensitive than the 7-day reproductive endpoint for Seridaphnia dubia. Though we didn't show results for bioaccumulation, what we found in the analyses of the tissue results suggests that tissue equilibrium is typically being realized by day 14, and this is consistent with what others are reporting in the literature. We also had bioconcentration factor calculations that indicate that there's a decreased BCF at the highest 2 exposures, 20 and 100 ppb, and this is potentially, overwhelming transport processes and active sites is a possible explanation for that. Another component that I didn't present as part of this work, but was also conducted as part of the studies, we looked at the influence of the chorion on exposures in embryos. Well, we exposed a series of embryos with a chorion on and chorion off, and what we found is that we did essentially get similar magnitude of impact, but the time to impact was faster with a chorion off in the exposed embryos. So slide 46. So in terms of benefits to Department of Defense, this study really helped sort of ground truth a key study that was driving regulations related to PFOS. It helped facilitate an improved understanding of mode of action and will help inform the ultimate adverse outcome pathway derivation. It improved derivation. It provides an improved basis for derivation of risk estimates. And one of the things that I haven't pointed out, but I will in the next slide, is this study was or benefited from the combined support of both SERDP as well as separate funding from private industry to leverage the work associated with this work. If I can have the next slide, please. I am just the spokesmodel for this work today. It involved collaboration and input from a lot of folks here at the ERDC, Dr. Kurt Gus and Natalia Venus, folks that really were key to the instrumental to this work, as included Eric, Milroy, and Ashley Kimball. And with that, I will wrap up because I'm over time.
Thank you so much, David. This is a great presentation. We've received a number of questions and we'll have time to get through some of them. Has the study actually had an impact on the derivation of regulatory levels in the United States or Australia?
Yeah, although we're sort of on the cusp of we have a manuscript basically completed, and it's not been published, but we're hoping to get it submitted either later this month or early the following month. But because of the work and our collaboration with EPA had situational awareness of the limitations of the Keter study, and basically elected not to include those results in the derivation of their recently released aquatic life criteria for PFOS. We've gone through some calculations to look at the influence of our results in a later iteration of the aquatic life criteria, and it's basically the value will not change. The EPA value will not change if our values are incorporated into their study. I have also spoken informally with representatives from EPA Victoria in Australia, and they are currently revising their 99% protection threshold from 0.0023 to looks like it's probably going to be somewhere around 9 parts per trillion. And though they're still including the results from Keter et al. For the time being, they did adjust the conservatism of their previous calculations based on the uncertainty associated with the Keter study.
Thanks so much, David. This question is from the Michigan Department of Natural Resources. Is the assumption that the fish absorb PFAS through their gills?
Yes, it's generally assumed that fish absorb PFAS across the gill interface and take it into their bloodstream. You know, as folks on the phone are probably aware, that PFAS are unique in their tendency to affiliate with proteinaceous material in the organism. So gills, blood, liver are key areas where PFOS tends to concentrate.
Thank you so much. This next question is from the South Carolina Department of Health and Environmental Control. What are the next steps for your research? Are you planning to test additional species? And are you going to do any more generational testing?
Well, I'll start with the last part first. I don't know that we'll be doing any more generational testing unless we find, you know, as we evaluate the additional PFAS that are out there, we find other PFAS that appear to be more problematic than PFOS, and it may warrant doing additional generational testing. Currently, we are looking at other PFAS constituents in both freshwater and marine species. And we're also looking at the association of PFAS in both freshwater and marine environments to sediments, and looking at bioavailability associated with PFAS in those sediment systems. So there's a lot of intriguing questions to be answered to try to understand how PFAS moves through the environment into organisms and through food webs. And those are some of the questions we're focused on.
Great, thank you. This next question is from Loyola University. Were the fish continually exposed to PFAS in between generations, and did you observe any PFAS effects on egg production?
So yes, we exposed the animals continuously over the 374 days of the 3 generations. We did not see any effect of PFOS exposure on egg production or egg viability.
Great, thank you. Given the complexity, costs, and potential risk of failure of longer-term multi-generational exposure, and based on your study, would you feel that this type of study is necessary and that the return on investment is there?
Yeah, so that's a good question. You know, in establishing ecological risk, we're generally concern ourselves with the viability of populations, which can be impacted by really subtle changes in survival, growth, and reproduction over multiple generations. I would say, while not for the faint of heart, these multi-generational studies do provide additional backstop for identification of subtle effects that might not be captured in more conventional single-generation chronic exposures. That said, I would caveat that they should be reserved for chemical or materials of special concern, those things that are highly persistent, bioaccumulative, and known to be toxic at very low concentrations. The use of a model like Danny Herrera, the zebrafish, does provide the additional benefit of elucidating mode of action and AOPs applicable across vertebrates, including humans, as we've got a good understanding of their genetics. So it is a good animal model to be able to do cross-species comparisons.
All right, and really quickly, one last question before we move over to Chris's presentation. Do you believe you generated enough studies, enough data in your study to develop a bioconcentration factor?
The short answer is yes. There is a publication that will be coming out in a couple of months related to the tissue residue information that was collected as part of the study, but not presented today. And so those values will be presented as part of that work.
And will you be including these results in your final report for SERDP and ESTCP?
Yes.
All right, so for those of you calling in, if you download the slides at the end of David's presentation, there's a link to his project webpage, and that's where the final report will be posted. When do you expect to finish the final report, David?
Oh, Lordy. So we have a series of manuscripts that are in prep currently. As I mentioned, the big apical endpoint study should probably be submitted to the journals next month. Then that'll fall in short order by the studies on bioconcentration, biouptake, and some of the subcellular effects, the histopathology, the telogen and the RNA sequencing information. I expect all of those manuscripts to be rolled up into a final report sometime this summer is the goal.
Great. Well, thank you so much, David, for a great presentation. We're going to go ahead and transition to the next speaker, but we'll make sure to save some time at the end to get back to any questions that we did not answer for you. So our second speaker today, thank you, David. Our second speaker today is Dr. Christine Custer, who's a research wildlife biologist at the USGS Upper Midwest Environmental Sciences Center. Since the 1970s, Chris's research has focused on the effect of contaminants on avian populations. She's looked at legacy organics, DDE, PCBs, dioxins, and furans. And more recently, she's evaluated other classes of contaminants such as trace elements, PAHs, and now PFAS. Species she has focused on in her studies have also varied depending on the geographic location, food web of concern, and other factors. Her most recent work centers on PFAS effects on tree swallows, and that's what she'll talk about today. She received her bachelor's degree in anthropology from the University of Colorado, a master's degree in biology from Corpus Christi State University, and a doctoral degree in wildlife and fisheries from Louisiana State University. Chris, we're delighted to have you. Please go ahead.
Okay, thank you so much for that introduction. And I just have to do a brief disclaimer that all the information I'm going to present today is preliminary. We still have more data to collect and more data to analyze. So just please keep that in mind. So the outline of my presentation today will be to introduce the study species, outline a few key questions, and then just present a snapshot of the preliminary data, and then finally some take-home messages, conclusions, and benefits. So I wanted to start my presentation just showing you this map of about 90 sites where we have been doing PFAS work since 2007. So the size of the circle indicates the concentration of total PFAS nanograms per ml in tree swallow nestling plasma. So there's a couple reasons to show this particular slide. One, we have a lot of information on PFAS and tree swallows. And this is important because if you want data and collect data from a new site, we have a ready-made database to compare that to many other sites across the US. The Canadian Wildlife Service also has been doing PFAS work. So there's a good database to compare to if you're looking at tree swallows. The second point I want to make about this slide is that there's a lot of background locations. So the size of the bubble indicates the amount of PFAS. The largest bubbles are greater than 600 nanograms per ml, but the very small dots are below about 200 nanograms per ml. So I think it's important because, you know, PFAS has been in use for several decades, and at least in birds, it's in most bird species globally. So you see several locations with quite large bubbles, and those have two sources. The Michigan site and the Minnesota site up by Duluth are primarily AFFF locations, but down near the Twin Cities, we have a location that has to do with a manufacturing plant. And then I'd like to call your attention to the medium-sized bubbles. If you look along the upper Mississippi River, but also along the Detroit River in southeastern Michigan and northern Ohio, but also bubbles over in the Niagara and Buffalo rivers in New York and the Chicago area, et cetera. So there's some urban industrial sources in some of these large urban industrial areas. So for tree swallows, we've got three major sources of different kinds of PFAS. And I think that's going to really help us understand what's going on with this very complicated group of contaminants. So our study design when we do a tree swallow study is to put up about 20 nest boxes at each of our sites or subsites. The pictures that you see here are a typical tree swallow clutch of eggs, and also a nest with the nestlings that are, one is just hatched and the one is in the process of hatching. So what we do is monitor those nest boxes weekly, sometimes more frequently depending on the chronology, and we quantify the number of eggs laid, the number that hatch, and the number that reach 12 days of age, which is what I'll call fledging. And then we use those data to look at and calculate reproductive success. So we collect our samples for chemical analyses as well as biochemical analyses at appropriate times. And of course, our two major matrices are eggs and these 12-day-old nestlings. Our chemical analyses are done by SGS Axis in Canada. And we've also had some trace element data collected from these same birds by the Terrell lab at Texas A&M University. And then all of our biomarker analyses that I'll talk about in a little bit are done by various collaborators, including Natalie Corona-Rainier at the Patuxent Wildlife Research Center and Cole Matson at Baylor. So our exposure endpoints, as I just mentioned, are in the eggs and also in the 12-day-old nestlings. And we have two kinds of additional endpoints, effects, which I'm reserving to talk about population level endpoints as well as responses. So at our population levels, we're looking at the hatching success, either the proportion of eggs that hatch or the daily probability of nestling survival. And we regress that against the concentration in those eggs to determine if there's like embryo toxicity for one thing. And then we also can look at and assess fledging success, which we do the proportion of the nestlings that hatch against the concentrations of PFAS in those same nestlings. And then we have 5 or 6 biomarkers that we've been looking at, and they range from liver enzyme analyses, this EROD assessment. We're looking at DNA damage in red blood cells, measuring thyroid hormone levels in the thyroid gland, And then we do oxidative stress. There's about 6 measures of those that can indicate cellular damage. And then also Natalie's developing some new immune functions analyses, and then we are dabbling in transcriptomics. So all of these endpoints are then regressed against either total PFAS or individual type of PFAS. So the key questions that I've set out to answer are PFAS causing adverse effects at levels that we're seeing in the environment? If so, what are those effects? Are they population level effects, or are they responses at a more cellular genetic level? And then if we're seeing effects, which of the PFAS are responsible? Because as this audience knows, there's many, many kinds of PFAS out there that animals in the environment are being exposed to. And then again, if those answers are positive, then at what concentrations are we seeing effects? Because as that map showed, there are PFAS in most birds out in the environment. And our key question is, that's fact. So we have to determine at what concentrations there are effects, if any. So the SERDP work, we're working on the East Coast at several Department of Defense study sites. And our field work was in 2021 and 2022, although we got started in 2020 to get our bird population set up and utilizing these areas. So our Department of Defense sites is we have 3 sites at Willow Grove in Pennsylvania. We have 2 sites at Joint Base McGuire-Dix Lakehurst. Our reference site for this particular study is the U.S. Fish and Wildlife Service Protectant Research Refuge outside of Laurel, Maryland. And then we also had a study site at the Naval Research Lab in Chesapeake Beach. But along with this work that we're conducting, we're also continuing our work at a manufacturing site, or at least the waste dump site from a manufacturing site in Minnesota. And then we also have data from some of these large urban and industrial corridors in Michigan, Wisconsin, and Minnesota. And then we were fortunate to also be working at some AFFF sites, the one near Duluth, Minnesota, the Oscoda, Michigan site, and then also we worked a couple of years at Cape Cod, Massachusetts. So I'm going to talk next about our exposure endpoints. Now, this table are the 10 or so PFAS that we detected in at least 90% or more of the eggs and nestling carcasses. And there's a couple of these PFAS, and I've indicated them, that were detected in slightly less than 90%, but I've included them on this table as well. Now, SGS Access is currently analyzing for 40 PFAS, so that's the number that we've been analyzing for. Now, I've color-coded these. So the 2 PFAS with the brightest green, the hexasulfonate and then PFOS, were the 2 chemicals that were in the highest concentrations in all of the historic AFFF formulations. The 2 PFAS with the slightly lighter green were present in historic AFFF formulations, but at much lower concentrations. And those are PFOA at the top and PFDS at the bottom. And then there's 6 PFAS that were detected in all the eggs and all the carcasses. And these were actually not ever present in the AFFF formulations. So I want to bring this up because it brings us back to that initial map that I showed, is that PFAS in birds is pretty much in all the bird species and pretty much everywhere. But at any particular site, you're going to probably have a mixture of PFAS that came, if you're at an AFFF location, from AFFF, but also PFAS perhaps from atmospheric deposition, or perhaps a metabolic byproduct in the birds or the insects. So, it's something we need to think about and figure out how to, if we're specifically interested in AFFF site, to subtract out those that weren't part of AFFF but had a different source. So, just a quick summary of our detection frequency is that in birds, we tend to see the longer chain carboxylates and sulfonates in the bird tissues. Some of these PFAS come from other sources or perhaps from metabolic or environmental processes that are occurring. And I'll talk a little bit more about this on the next slide or two, but the hexasulfonate, which is the second most common PFAS in those historic AFFF formulations, could be a quick and dirty indicator at a site if you actually have much or any AFFF contributions to the PFAS in the birds at that site. So what you have in these pies is the proportion of total PFAS in tree swallow nestlings that are comprised of the different PFAS. So the far left pie is from an AFFF site at Willow Grove, Pennsylvania, and the mean concentration at that site was 97.6 nanograms per gram wet weight in those tree swallow nestlings. So what we found at that site was a little over 75% of total PFAS was comprised of PFOS, that 8-carbon sulfonate. But we have a sizable wedge based on the information that I just talked about, was sizable wedge for the hexasulfonate. And then there's a little bit of the PFDA, the 10-carbon, and PF-dodecanoate, the 12-carbon carboxylic acid. And then 15% or so are all the other 29 PFAS that we also detected in those nestling carcasses. Now, I want to compare that with one of our manufacturing sites in the Twin Cities, Minnesota, which had roughly the same amount of PFAS, total PFAS exposure, 106.3 nanograms per gram wet weight. But you can see that the makeup of those PFAS was somewhat different. So you've got maybe 80% of that total was PFOS. Note that we didn't find any hexasulfonate. There was just a little bit of the decanoate, and then again, roughly the same proportion of all the other 29 PFAS. Finally, the pie on the far right was our reference location at the Patuxent Research Refuge, and we had about 20 times less PFAS at that site, 4.5 nanograms per gram wet weight, but a very different look at what kinds of PFAS you find in those nestling carcasses. So PFOS comprised less than 50% of the total. You have quite significant wedges of the decanoate and the dodecanoate, the green and the salmon color. You've got a whole lot of everything else there, which I think is kind of interesting. And then you have no hexasulfonate at that site. So at our non-AFFF sites, there's little or no hexasulfonate in these proportions. Now, another type of data we have, of course, is just our actual concentration. So this is total PFAS in tree swallow nestling carcasses from just 2021. So on most of the slides that you'll see, our sites are going to be along the x-axis. So on this slide, we have our 3 sites from Joint Base Willow Grove, 2 from McGuire-Dix Lakehurst, then our Naval Research Lab, our Cape Cod site, and then our 2 reference locations. And then along the y-axis is actual concentration in those nestling carcasses, nanograms per gram wet weight. The horizontal red bars are our median, our geometric mean, and then our confidence intervals. So for this particular year, the Wreck Pond and Runway had slightly less PFAS exposure than our 2 sites at Dix and Lakehurst. Our Air National Guard base, we're continuing to work at that site, seems to have a bit higher and more similar to the concentrations we found at the Naval Research Lab at Chesapeake Beach. And then we have another jump again in exposure to about 300 nanograms per gram wet weight in those carcasses at the Ashumet Pond just off of Cape Cod, Joint Base Cape Cod. And then all of these sites had significantly higher concentrations in tree swallow nestling carcasses than at our Patuxent Research Refuge site. And then I also sampled another site on that refuge, which was just off Tipton Tipton Air Base, which is just off of Fort Meade. And this was just to give the refuge a bit more information about what's happening on their property. So what you see on this table then is the total PFAS concentrations, again, nanograms per gram wet weight in the tree swallow diet. And these are insects that we took out of the stomachs of the tree swallow nestlings that we euthanized. So at Willow Grove, the top two lines in diet, we had anywhere between about 15 and 30 nanograms per gram. Our Patuxent Refuge site, which you remember was significantly lower, had between 4 and 5 nanograms per gram in those stomach contents. And if you jump to the very bottom, our North Track, our other reference location, was about 8, so very consistent. But if you'll remember from the previous slide, there was a small increase in the amount of PFOS in the nestling, or PFAS in the nestlings, and that's reflected in these diet, which are again twice as high as at Willow Grove, and then Cape Cod, which was again even more exposed in the tree swallow nestlings, had higher concentrations in those diet samples. So I'm bringing this up because if you take the average concentration in the nestling carcass and regress it against those diet concentrations, we find that the R is 0.98, which is truly phenomenal. So for these birds, it's all about what they're consuming. It's all about what's in their food and the types of, I've shown pictures of the types of food that they're tend to eat at the bottom: mayflies, dipteran, caddisflies, etc. So there could be some contribution from air inhalation or water consumption, but for these birds, that contribution, I think, is very minimal. So I put this out as a bit of a cautionary tale because concentrations in water and perhaps composition of what's in the water, and I'll show that on the next slide, may not be all that useful for predicting what types of PFAS we're going to find in the biota at a particular site. So I'd like to thank Wes Flynn for allowing me to use some of his data from a tannery waste site. So again, this is proportion of the different types of PFAS that comprise total PFAS. That's on the y-axis. And then the two leftmost bars are his two abiotic endpoints, the different types of PFAS in the water and in sediment. And then to the right of that black line, we have two bars that are algae and aquatic plants. And then the rest of the bars are what we're seeing in biota. So PFOS is that kind of purplish blue bar. And what I want you to take home from this is that in water, that was hardly detected. It was a very small proportion of the total, but it makes up 75 and higher % of what we're finding in the animals, except for the amphibians. And Wes will have to explain what exactly is going on there. So the sediment, though, seems to be a better predictor in terms of the proportion of the different types of PFAS that we're finding in biotic tissue is more similar to what we're finding in the sediment. So I'm going to just talk a little bit and just give data on a few of our effects endpoints, but we have more of these than I'll have time to show this afternoon. So the first one is one of the most important, I think, on a population level, is the hatching success. So the data in the table is for our 2021 data. Now we have average % hatching at our 86 sites across the Great Lakes. That was the map that I showed earlier. And at those 86 sites, the average hatching success is about 75% of the eggs laid actually hatch. So that's kind of the bar that we want to either be at or exceed. So on this table, the center column is the number of nests, and then the % hatching is the column on the far right. So at our 3 sites in Willow Grove, hatching success was between 78 and 100% of the eggs hatched. At Joint Base McGuire-Dix Lakehurst, 92 and 95% of the eggs hatched. Our Patuxent Research Refuge actually had the lowest % of eggs hatching, 68%. But I do want to note that we had some weather issues in 2021 early on, and I still need to factor that in. So I believe that reproduction there will be slightly better than it's showing here. But at our North Track reference site near Fort Meade, we had 80% of the eggs hatched, And then at Cape Cod, with the highest concentrations in 2021, 79% of the eggs hatched. So it would appear at using, looking at these data that the hatching success appeared normal, but I do want to put out the caveat that we've got more refined statistical tests that we're in the process of doing to use logistic regression, the amount of contaminants in that clutch versus the hatching success of that clutch, as well as regressing against the daily probability of egg survival and failure. So we've got more to do, but I don't see red flags at this point. This is just one of the many biomarkers that we're looking at, but it was one that could potentially be affected by PFAS, and this is immune response metric. It's acute phase protein, HAPT, which is on the y-axis, and then again, our various locations are along the x-axis. And if you have a toxic or a disease challenge, a pathogenic episode at a site or a toxic challenge, then you would expect that these acute phase proteins are released from the liver. So at Willow Grove, Dix, Lakehurst, Chesapeake Beach, and Cape Cod, those concentrations are very similar, a few higher, but a few lower, and a few just the same as we found at our two reference locations, Patuxent and North Track. But they're also, we're seeing similar levels of this HAPT at some other types of PFAS source, our Gibson City, which is a food processing plant, and then two of our sites in the Twin Cities potentially affected by the manufacturing plant and just general upper Mississippi River industrial urban sources. So my conclusions are, I think we're well positioned with a matrix of sites with many different types of sources to answer the questions that we posed at the beginning. Are we seeing effects of PFAS at environmental levels on this model species, the tree swallow? And if we're seeing effects, what are those effects? Are they population level effects or are they biomarker responses? So far, but like I said, we have much more data to analyze in a much more sophisticated fashion, not seeing any red flags to date. And I'll just put in a plug for tree swallows. I think they're quite an easy species to study. They've got a lot of benefits in terms of sampling real world exposures and effects. And I think they could be used at many other Department of Defense sites, at least across the northern half of the US and into Canada. You know, they don't breed in the southern tier of states, at least very frequently, like in Florida and Texas and, you know, a lot of the southern states, but they're a great species in the northern half of the US. So I think some of the benefits of this work to the Department of Defense is we have excellent context for the data. So if you are collecting a similar data on a similar aquatic insectivore like the tree swallow, can be compared to multiple sites across the US and also across Canada. I think having many different PFAS sources for comparative purposes, I think are gonna help us elucidate if we're seeing effects exactly which PFAS, because you know, some of these PFAS we are not finding at AFFF sites, but we are finding at other types of source sites. And then I also think looking at different levels of biological effects, and this is similar to what Dave did, a lot of different kinds of endpoints can help us much better understand effects and responses and potential effects out in the environment. So I'd like to thank Paul Dummer with my office in USGS in La Crosse, Wisconsin. Also Natalie Corona-Ranier, who's with USGS in Laurel, Maryland, and is responsible for many of the biomarker assessments and endpoints, as well as Cole Madsen, who's with Baylor University, and he's been responsible for the genetic damage and transcriptomic assessments. So there's my contact information, my email, as well as a phone. And again, as for all of these, you can go to that SERDP webpage. And there's not a lot of data and reports there yet. I anticipate in about a year, the final report will be through USGS review and posted there, but looking forward to to working through the data and just seeing what we've got going.
Great,
So,
thank you so much, Christine. Great presentation. We're going to go ahead and start with the questions that have been coming in. You are looking at 5 or so biomarker analyses. Would you expect that PFAS would be affecting all of these biomarkers?
and the answer to that is no. So for instance, let's take EROD. That's a biomarker we've been doing for about 20 or 30 years. And that biomarker is enhanced when you have exposure to PCBs, PAHs, dioxin, and furans. So we're keeping that in this particular study because we're talking about a natural system, and we don't actually know everything that could be, say, at any of these Department of Defense sites or even at our reference location. So had we seen, or if we see EROD induced, then we know to delve a little bit further, have additional samples analyzed for some of these chemicals that we know can induce EROD. So it's somewhat of a check on ourselves that when we start asking the question, if there's a reproductive effect, is it due to PFAS? Or if we have EROD induction, then we need to say, well, maybe there's some influence of PCBs or dioxins. Another one that we don't expect to have associated with PFAS necessarily are the genetic damage endpoints. Those are associated with clastogenic compounds like some of the trace elements, but also radiation. So again, we don't expect that one to be affected, but it would clue us in to look a little further if it was. But some of the immune response, the transcriptomics, et cetera, and perhaps the cellular damage endpoints, the oxidative stress, could well be associated with PFAS. And that's one of the questions we'll be answering with our data.
Thank you, Chris. This next question is from Loyola University. Do you know if the abiotic versus biotic distribution among species follows PFAS production?
Okay, I'm not 100% sure I understand the full part of that question, but at sites, an AFFF site, I would anticipate that yes, you'd have more exposure in the birds compared to a site with, just background exposure or atmospheric deposition. But what this particular SERDP project will do, along with the various collaborators, will allow us to answer that question more closely, because at Willow Grove, for instance, there's good water data, there's good sediment data, there's data from fish and data from invertebrates. So we should be able to build that food web model, using some of these data. At many of the other sites we've worked, we don't have that abiotic backup. Now, some sites we do, and we'll be looking into those, but at many of these sites, no one knows how much PFAS is in the water or the sediments at those sites.
Thank you, Chris. This next question is from the Oak Ridge National Lab. If local water concentrations of the carboxylic acids, or PFCAs, are not indicative of PFCA concentrations within eggs and nestlings, how are the PFCAs entering the food chain to magnify within tree swallows?
So I'm not a physiologist, so there'll probably be better people to ask that question of. But sometimes, especially in the abiotic media, those concentrations are so low that they're below the detection limits. So you've got a water sample that's below the detection limit, but it may still be there. So by the time you bioconcentrate and biomagnify some of these, then all of a sudden in the aquatic insects, you'll sometimes start to pick it up, and then in the birds, you'll pick it up. So partly, I think it's a detection limit issue that it says, well, it's not in the water, but oh my goodness, we're seeing it in the birds or their food. So I think partly, as the detection limits get better, we'll have fewer of those. But I think as we analyze our data, and especially with the Willow Grove, we should be able to look at water to insects to birds and then perhaps suss out a better answer to that question.
Great, thank you so much, Chris. Were there any differences in plasma PFAS in male versus female birds? Was that something that you looked at?
We should be able to do a little bit of that. We have the sex of the tree swallow nestlings, thanks to Natalie and her collaborators, and then we'll have PFAS in those nestling plasmas. So we should be able to answer that question, but I would not at this point anticipate a difference because all our data from nestlings are only 12 days of age. They haven't been sexually differentiated too terribly much, and they're not in a reproductive status. The difference often for other kinds of contaminants between males and females are because the females are able to shunt a lot of that particular chemical into the egg and get it out of her body tissue, whereas the males do not have that elimination process. So for nestlings, I wouldn't anticipate a male to female difference, but certainly at the end of egg laying, I might anticipate a male to female difference in adult birds.
Great, thank you so much. A few more questions for you, then we'll pull David back in. You recommend the use of tree swallows as a model avian species at other DoD sites impacted by PFAS. Are there any downsides or complicating factors that you would like to discuss?
So I think it's a great endpoint, but then of course I'm biased on that regard. However, it's not a difficult endpoint to use, but you have to be proactive. So if you want to start studying tree swallows and it's now the middle of May, you're not going to pull it off this year. It's just too late in the season. So you have to be proactive. You have to know where you want to do a study. And my preference is to get those nest boxes in later on this fall. And then when the birds first arrive next spring in the end of March, they're able to find and colonize those boxes. So that's one downside. You can't just decide tomorrow that you're going to do a tree swallow study unless you already have a series of boxes out, which mostly you don't have a series of boxes out. And, again, there's some logistic issues. You have to have a federal collecting permit for a migratory bird. You've got to have state collecting permits. Sometimes if you're at a park or wherever, you've got to have local collecting permits. So there's a lot of logistic issues that you've got to deal with. None of them are hard, but it's not like deciding that, geez, I want a water sample from this location, and you can grab your sampling gear and go out there tomorrow and sample. So you've got to plan a little bit farther ahead, but I do think the benefits outweigh, some of these potential issues, because again, it's, sampling the real world and exactly what these birds are being exposed to and in what quantities.
Great, thank you. And one last question for you from Parsons. Do you have plans to develop a data regression of PFAS concentrations in diet versus measurements of reproductive success?
Yes, we can certainly do that. It's not going to be a very robust regression because these diet samples come from the stomach of our tree swallow nestlings, and there's not much food in each individual nestling. So all of our diet data have to be composited by site. So all of a sudden, you're down to your 7 or 8 sites that you're actually sampling. So it wouldn't be a very robust regression, which is why we'll be regressing concentrations in that clutch of eggs against the proportion of eggs that hatch. And that'll be, much larger sample size, and I think a much better test. But we can certainly do this smaller regression, but our power will be less just because of the smaller sample size, but certainly something we can easily do.
All right, and one last question. Your data showed differences between between avian responses at the different installations you looked at. What do you think accounts for these differences?
So for the biomarker responses, it is just normal variation. That's one of the, I don't call it necessarily an issue, but there's always more variation in our biomarker response mechanisms, i.e. our 95% confidence intervals are a little broader. And for our exposure endpoints, again, we have a level of variation. And those 95% confidence intervals are going to shrink down as we incorporate our 2022 data, and have many more samples, and our confidence intervals get much smaller. But again, it's just normal variation, which I think is important because each individual bird reacts differently, has slightly different metabolism, perhaps feeding maybe on a slightly different part of a river stretch or whatever. So, you're always going to have that normal variation that you have to contend with.
Thank you, Chris. At this point, we have a few more questions that came in for David. So I'm going to ask David to come back on, and then we'll conclude with additional questions for Chris. David, how does PFAS elimination via eggs affect the exposure difference between male and female zebrafish?
That is an excellent question, and one that we're actually looking to quantify. We do believe that it is explaining some of the differences we're seeing between males and females is the elimination, maternal elimination through egg production. Hopefully, as I said, that analysis is underway, and we're hoping to include it as part of our future publication to describe the kinetics of that.
Thank you. Next question for you. Did the zebrafish study account for non-toxicity effects of PFAS that could also pose a health effect on the zebrafish?
Not sure that I entirely understand the question. If we're thinking about sort of like maybe water quality parameters and those kinds of things that could have influenced the outcome, indeed, those were monitored over the course of the exposure, and everything was within, effective control limits for the health of the species. So of the things that we were able to monitor, we didn't see anything untoward that would have resulted in those effects. And I would say also, while the study was focused on PFOS, of course, we used essentially the same analyte list that's in the current 1633 method. So we were looking for, I think, on the order of about 44 different PFAS analytes, and PFOS was the only one that was measurable at very low quantitation limits. So there wasn't anything else related to PFAS in the system that could be influencing the results of the study. So I'm not sure that I'm answering the question, but.
Well, thank you for taking a crack at it. Both you and Chris alluded to gaps that you're going to be addressing. Are there any existing data gaps that you believe should be addressed that you haven't talked about yet? We'll start with you, Dave, and conclude with Chris.
I think in looking at Chris's work and the relationship between sediments, the distribution of PFOS or PFAS in sediments versus what she was measuring in blood plasma, that correlation is obviously intriguing. Whether it is a consequence of feeding on organisms that spend their life in the benthos and then emerge and then get consumed by the swallows is something that we need to understand what the driver is from an availability standpoint in sediments, and whether sediments can serve as a source of environmental exposure. So that's one of the areas that I'm very interested in trying to get my arms around for PFAS more generally.
Thank you so much, David. And really quickly, Chris, is there anything you'd like to add?
No, I think from my standpoint, what I need and would like to do would be dig a little bit deeper to find, a lot more of the PFAS data that are out there in state databases and various places, for these abiotic metrics, both water and sediment, to see if there's any more data that we could tie in with our tree swallow model at not only the sites from this study, but our previous sites, to do a little better of looking at this food chain, how these PFAS are moving through the food chain. And then, outside my realm, but certainly other bird species that have a local distribution, and especially birds in that fish, the piscivores, and how some of these assessments in terms of accumulation, might be different depending on the food web. And in fact, we're actually doing a joint project with Jamie Susky and Chris Solis to look at a terrestrial insectivore, the house wren. So we're going to be trying to fill a gap on how some of these other ecosystems right there around a PFAS source might be affected and being exposed to the PFAS. How far away from that source, perhaps, are we having significant PFAS exposure? So we're working to fill one of those gaps with a new SERDP proposal that's getting kicked off this year.
Thank you so much, Chris, and thank you again, David. We're going to go ahead and wrap up with a reminder about the next webinar on June 1st, which will focus on predictive corrosion models to estimate the functional life of protective coatings on various DoD assets. We'll have two speakers, Dr. Justin Biffinger from the University of Dayton and Dr. Charles White from the Naval Surface Warfare Center. Registration is open, so please visit the SERDP and ESTCP webinar webpage to register for this and other webinars through the beginning of next year. Before we conclude, I'd like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you'd like to refer to them in the future. We would appreciate it at this time if you can take a moment to complete the survey that will pop up on your screen. This concludes today's webcast. Thank you for joining us.